Gene editing is a field of editing or genetic code like programming software. The technology allows us to make fixes or enhances to the DNA of anything that's living. By anything, I mean everything with a genetic code ranging from plants and fruits to animals and even humans. Genetic code takes the form of what's called a double helix. Each side of a helix has a letter of A, C, T, or G. The combination of these is what gives us certain features like hair type or eye color. CRISPR is a natural process that was originally meant for defending bacteria against viruses. It uses two main components: short snippets of repetitive DNA sequences called clustered, regularly interspaced short palindromic repeats. We have CRISPR-associated protein, which chops DNA like scissors. In CRISPR RNA is a sequence of DNA that can be taken from a virus. It binds to a protein called Cas9, and it acts like a security guard and scouts until it finds an exact sequence of DNA, and then it destroys that DNA. We can use this mechanism to target DNA we want, to cut it, and replace it. How we replace it is we give it a template DNA, and when it's naturally repairing it, it'll repair itself based off that template. This technology opens up so many possibilities in the future. We can make cure or treatments for diseases like sickle cell anemia, HIV, cancer, and more. We can do much more than cure disease though. We can even make designer humans. Imagine customizing features like eye color, height, weight, or even our health span just by tweaking our genes. The possibilities are endless. A big problem with this new technology is the ethics behind it. Some examples are the technology is rather too expensive to use. Does it mean that only wealthy people get to use it? Targeting people with a certain RNA as a bioweapon, and most of all, using genetic engineering is like playing God, and the worst part is, unlike accidentally deleting a paragraph from an essay, there's no undo button. You might be thinking, well, the only way to prevent this is to stop the research, but even if we make it illegal, it's better to do the research and create regulations and ways to protect ourselves. Now, let's talk about Inscriptia, a company that's committed to pushing the research forward. They raised a total funding of $309.6 million. They're developing a unique family of CRISPR enzymes called MAD enzymes. They made the license for the MAD7 enzyme free for research. Their other product, Onyx, takes months to edit genes rather than the normal way, which takes years. Onyx is the only platform that makes scalable digital genome editing possible. This will change and impact the future greatly. Thanks for watching. I hope you took something out of this, and I hope you're excited for the future of this technology.